Hey everyone, I am going to present over Enterovirus D68, which is a non-polio enterovirus. And in this presentation, I'm going to talk about some of its characteristics and its pathogenicity, along with some epidemiological things that I have discovered in my research. And it is a member of the Picornaviridae family. And among this family are many genuses that include viruses such as the hepatovirus genus, which contains hepatitis A, which we have seen in class already. And then the genus of my disease is Enterovirus genus. It contains 7 different species, plus some animal species that I haven't put on here. And these species are the rhinoviruses A, B, and C, and then the enteroviruses A through D. Enterovirus D species contains our serotype EV-D68, and this is sort of a mixture between the rhinoviruses and the enteroviruses. So rhinoviruses are associated with common cold usually, and then enteroviruses are associated with gastrointestinal infections. And what's interesting is enterovirus C contains the poliovirus. So my disease is sort of a mixture between the polioviruses and the common cold. It is composed of a non-enveloped icosahedral capsid, which you can see here. And then inside this capsid is positive stranded ssRNA. And the way it affects the human is it is transmitted into the upper respiratory tract through somebody sneezing or inhaling water droplets that contain the virus. And what happens is the capsid binds to an alpha-2 linked sialylated receptor, which here is a neuramic acid, which is a sialic acid that is found on epithelial cells. And these form chains from the 2 carbon here to the 6 carbon of galactose. And these are alternating chains of the neuramic acid and the galactose. And these are on the outside of receptor proteins. So to visualize it, here are the chains of carbohydrates, and then the receptor protein that is sialylated. And from here, the viruses are Pac-Man, and this virus binds through a cleft that attaches to sialic acid. So these clefts are seen here in blue. And so these clefts bind the sialic acid. This induces a conformational change in the capsid, which ultimately results in an injection of a hydrophobic residue along with a fatty acid chain that goes into the membrane. And from here, it triggers the collapse of the capsid proteins. And the mechanism hasn't really been discovered yet, but either through endocytosis or injecting the genome directly, it enters into the cell and then can reproduce. So then some consequences of this infection is that it results in a cough or difficulty breathing. So many of these symptoms are similar to the common cold. So a lot of people may not go seek treatment, but for some children and teenagers, this can result in severe respiratory issues, and some even have weakness in the limbs similar to poliovirus. But the correlation between the virus and this weakness has not been found yet. It is usually diagnosed through PCR, and the main problem in the clinical world is that most clinics cannot differentiate it between enteroviruses and rhinoviruses. So if EV-D68 is expected, then they have to send their strain off to the CDC where they have an actual test for ED or EV-D68. And then from here, there's not many treatment options for the disease. So they mainly just treat the symptoms because no vaccine or antivirals have been developed since an animal model has not shown to be really useful in researching the disease. And these treatments that improve the symptoms include corticosteroids and bronchodilators, which open up your lungs and allow you to breathe a little easier when you have the infection. So to get behind some of the epidemiology behind this is first to look at the transmission of the disease. And it is transmitted through contact with respiratory secretions. So that's saliva, mucus, anything that comes out of your mouth pretty much if you're infected with it. And then the incubation period after infection is 2 to 3 days. And from here you can shed the virus for up to 3 weeks. And what's interesting is it can be harbored on environmental surfaces, as it's fairly stable in the environment. But since it's a virus, of course, it'll break down eventually. Then from here, we'll look at some history from the virus. So it was first isolated in 1962 by a pediatrician in California. And then from here until 2005, there were not many cases of the virus. Only 26 were reported. And since 2005, there's been sporadic outbreaks of the virus pretty much worldwide. And an important one was the 2014 outbreak in the US, which infected over 1,200 people that needed medical care. And it's assumed that there are more that were infected, so upwards of 5,000 people that just did not seek treatment because of their symptoms were mild or they're carriers of the disease. And so what's interesting here is that there must have been some genetic mutation or increase in variance of the disease between 1970 and 2005. And a lot of research has been undergone to discover this, but they still don't know the cause exactly. They know there are variants in some of the capsid proteins that might have affected the virulence of the virus. So this may have led to the outbreaks that have been more recent. So future outlook of the virus is that since it has become more virulent in the last 50 years, it's necessary that we research it more just in case it does continue to become more virulent and to mutate into a more pathogenic virus. But since it only causes mild symptoms in adults that are immune to it, it's not like great desire of the medical community to research it. But since the 2014 outbreak, research has been sparked on the virus. And the main limitation to this research is that there's not a good animal model. But I have seen articles that have used cotton mice to model the disease, and this might be a potential model in the future. So here are my references, and if you have any questions, I'm happy to answer them.